Thank you very much. Good afternoon, everyone. I will introduce you to ImmPACT Bio and the technologies within the company in the cell therapy space. The focus of the presentation will be on our clinical stage assets, but we do have a pipeline behind it I'll briefly mention. So very quickly, we are developing innovative CAR T-cell therapies with the focus on durability and persistence of the CAR T-cells in both the oncology space as well as autoimmune space. And as you'll see in our lead asset, anti-CD19, anti-CD20 CAR T program, we have very long-term persistence of the CAR T-cells, and we're having durability that actually exceeds over 2 years to 3 years now in many patients. So we are focused on a platform that's just not CAR-based, but we focused on cell therapy manufacturing, and I'll mention what that is in the slides also. We view our lead asset as a pipeline and a product, and even within the pipeline, we have a TGF-beta platform that can generate even more assets and more products from that space. We have in-house manufacturing. We focus on a CD62 ligand isolation of cells early on, and that really predisposes the product to be much more central memory population. And that's what gives it the advantage, I think, in the tumor activity space, as well as persistence. And we have seen that also, we've seen that as very beneficial in the autoimmune space due to the positive tolerability profile. We have advanced clinical programs. We have a phase 1 study ongoing in aggressive B-cell malignancies, B-cell lymphoma, and also we're about to start a study very soon in lupus nephritis and SLE, a phase 1-2 study in that space with the same program. So this is the pipeline, and this is the last time I'll mention the one in the preclinical phase. That's at the bottom of the slide. But we've basically taken a leverageable asset that was licensed from UCLA. So we have several assets from UCLA with Dr. Yvonne Chen and Dr. Tony Ribas. And we've taken the lead asset, an anti-CD19, CD20, and split it into 2 different programs. One is IMPT-314 and IMPT-514. IMPT-314 is for the autoimmune, for the oncology space, for heme malignancies, and IMPT-514 is for the autoimmune space, starting with obviously lupus nephritis and SLE, but able to expand into other areas where a anti-CD19, anti-CD20 dual approach makes sense. As I mentioned, the key milestones that we're expecting from this over this next year is an end of phase 1 meeting at the end of the first half of 2024, phase 2 initiation in the second half of 2024 for IMPT-314. On IMPT-514, phase 1 initiation, in fact, the IND and FTD, IND is cleared and the FTD has been granted, and we'll be activating sites at the end of this year, hopefully enrollment by early next year. And then just as a soft mention is our switch receptors, a TGF-beta in combination with Claudin 18.2 for solid tumors. We're focusing on the immunosuppressive tumor microenvironment, and we're actually starting with the upper GI cancers as an example. That will have IND-enabling studies complete in the first half of next year with an IND plan submission in the second half of next year. So we're going to focus now on the clinical asset. It's a CD19, CD20 directed bispecific CAR. It's a tandem CAR, so it's not a bicistronic CAR. Because it's a tandem CAR, it's quite efficient in terms of its construct size and CAR transduction and expression on the target cell and the T cell. It is developed empirically by Dr. Yvonne Chen, as I mentioned, at UCLA. She tried many, many different designs. It has the earliest patents in this particular area and very specific for how it binds the CD20 antigen and then the CD19 antigen and the dual binding. It is a true OR-gated CAR, so if you actually lose either antigen, it still works equally well. If you lose both, it won't work. It's very targeted. Okay, so if you have both, works. If you have one, it works. We have a 41BB co-stim domain that was the preferred choice for Yvonne when she developed this, and this is the platform that we move forward with. It has a gentler profile and also increased persistence of the CAR T cells. It also has a flexible linker in there. That flexible linker actually gives a uniqueness to this. It actually has the ability to bind. The CD20 is membrane distal for a reason. It really needs, the CAR needs to be further out from the membrane of the effector cell. So by putting a flexible linker, it can bind both 19 and 20. So the other part of the platform that's very important There is a CAR in there, of course, but the cell is very important. We made a very strategic decision to actually focus on cell types that have more stem-like qualities associated with it. Now, we didn't do that blindly. There's more recent data today that shows that it actually makes a difference. It reduces exhaustion, and it improves persistence. And this is the Yescarta data that has come out between 2020 and 2023, with clinical data showing that the only difference you'll see in patients that were treated with Yescarta, and when they looked at median for naive T-cell phenotype for high or low, that was the difference in the Kaplan-Meier curve. So it does make a difference which cell type ends up in the product versus a very high effector population or effector memory population versus central memory or naive population. I'll show you some data in that. So with that in mind, we start our manufacturing with CD62 ligand isolation, and that then, through the manufacturing process, predisposes the product to that central memory population. On the right-hand side is actually our own data across multiple healthy donors, SLE, lupus nephritis, as well as in lymphoma patients. And you see very, very consistently that our product is very predisposed to central memory, some little bit of effector memory, and then some naive cells in there. Very, very little effector cells are noted, almost all T cells. So the profile has been very consistent in this way. So what does that translate to in terms of clinical data? So on the B-cell malignancy side, what you have seen in the UCLA study was an investigator-initiated study at UCLA that shows that overall survival has not yet been reached in these patients. It's a cohort of 11 patients across a variety of lymphomas. And then the median PFS in the update that was provided at AACR earlier this year, it's 18.2 months, but there'll be an ASH cut. There were several patients that were less than 18.2 months, and they continues to stay in CR. So you'll see an ASH cut that'll show that the median progression-free survival continues to be very robust. Very importantly, because I think of the various attributes of the CAR with the 4-1BB, with this heavy central memory population, you're seeing no ICANS at all in the oncology setting. It's actually a pretty low dose, between 50 million CAR-positive cells to about 165 million CAR-positive cells. So lower than most of the commercial products are being given, but highly effective in efficacy. And you're only seeing grade 1 CRS or no CRS at all. Okay, so and you've seen this actually in the patients we have treated very recently with our own product, a very similar profile, no ICANS and grade 1 CRS. So the good part of this about this is it can become an outpatient therapy if this profile continues holding up. Very few cell therapies have this kind of profile in the oncology setting. So we are right now, as I mentioned, in a phase 1 study. We are starting with 100 million CAR-positive cells, and that can be dose escalated if we want to 300 million CAR-positive cells. We're doing both CAR-T experienced and CAR-T naive, so they've either had CD19 CAR and failed, or they have never had CD19 CAR. We have both cohorts in the phase 1 portion. We have to determine at the end of phase 1 meeting, based on the data, which population we take forward. We believe CAR-T experience is a very high unmet need right now. And then, of course, moving into second line with this favorable efficacy and safety profile are going to be very important. As I mentioned, this particular program actually has a fast track designation. So we'll move on to the autoimmune space. So why would we take something that's anti-CD19, anti-CD20 forward into autoimmune? And you'll see that the data is very compelling in this area to target both antigens. So number one is the B-cell and T-cell compartment. If you look at the B-cell compartment, there are very specific types of B-cell called age-associated B-cells. They're highly inflammatory in nature. They sit in end organs. They produce a lot of autoantibodies and immune deposition disease in these end organs. And this is one, for example, that helps destroy nephrons in the kidney. Okay, so if they express very high CD20. Okay, so they do express CD19 at variable levels, but CD20 is consistently expressed at a very high level, and they are one of the main producers of autoantibodies. We are importantly not targeting plasma cells. We believe that by targeting plasma cells, you may also wipe out the cells that are beneficial. You know, most of our long-lived plasma cells are based on all the vaccines, previous infections, and everything else that we have gotten. We don't want to lose that type of long-lived immunity in these patients, especially in autoimmune patients when they undergo several cycles of immunosuppressive drugs. But there is an emerging literature that's very important to note. There are certain types of dysfunctional T cells that express CD20. Now, I'm a B-cell biologist at the time when CD5 was discovered on B-cells, and it turned out that CD5, a T-cell antigen, was expressed by B-cells. It turns out to be predominantly in CLL and a few autoimmune diseases. Now, in T cells expressing a B cell antigen, what's going on here? So it's not quite clear if this is primarily due to mRNA transcripts and then CD20 being expressed on the surface, or it is actually because the T cell engulfs a target cell and then expresses the antigen. Both have been postulated, and mRNA transcripts have been shown, but we also see that without mRNA transcripts, T cells are expressing this. These are high producers of pro-inflammatory cytokines. They seem to be involved with the disease pathogenesis, and you can see here all of the various pro-inflammatory cytokines that have been associated with this CD20 expressing T cells. So 19 has recently emerged as a very exciting target in autoimmunity. But anti-CD20 approaches have been around for over 2 decades. I mean, anti-CD20 therapy has been utilized in autoimmune diseases for a long time, mostly rituximab, but several others are now entering into spaces like MS and other areas. So it's very well established in autoimmunity, and 19 has been very exciting to see the cell-based approaches coming out of the data in Germany, and now several other companies will be coming out with anti-19 approaches. But our dual approach, we believe, will have a deeper and more prolonged impact in terms of B-cell depletion, as well as the T-cell compartment that I just mentioned. So we actually got samples of patients with SLE and lupus. These are donors. And before we filed our IND and got it cleared, we wanted to make sure we can make effective product from these patients who were donors. And we showed very high transduction efficiency, comparable fold expansion. And remember, these are patients who are also on ongoing immunosuppressive drugs. So we want to make sure that the cells can still expand and you can make clinical dose. And we had very good fold expansion. We showed that it has very potent B-cell depletion activity, and we looked at a subclass of B-cells with IgG production, and we also had potent depletion of IgG-producing B-cells. Importantly, on the right-hand side, you'll notice something. The gamma interferon production from cells made with our product is far less than what you see with other approaches. And we have tested a Yescarta-like construct, a Breyanzi-like construct. We have tested these various constructs and how they're generally manufactured. We can't replicate those exactly from other companies, but our interferon production is far less, and we believe this may be one of the reasons, and this was actually seen also at UCLA, is one of the reasons we're getting really low-grade CRS, grade 1 or none, and no ICANS at all, because there's no initial cytokine storm when the patients are given the product. So what are we going to do here? We're starting with a refractory active lupus nephritis. We actually have it also open for extrarenal SLE. Once we establish dose and safety in this, we believe 50 million CAR-positive cells. Remember, 100 million in oncology, 50 million in autoimmunity. We're starting with less dose. The FDA will see our oncology data before even our autoimmune trial, because we're already dosing in the oncology side, and we will expand this. There are other potential indications where the anti-CD19, anti-CD20 approaches make sense. This is just a small list of the various approaches. As you know, in autoimmunity, there's over 100 B-cell mediated autoimmune diseases. Some of them are very small populations, and some of them are very significant. But all in all, the autoimmune space is far larger than the lymphoma space, and there will be more than enough patients out there for several companies to be successful in hopefully long-term drug-free remission for these patients. Again, I mentioned tolerability. Chronic diseases, I think, will actually have table stakes for having outpatient therapy. These are patients who are treated with multiple drugs. They get hospitalized for flares, and I think there'll be a benefit of going outpatient in the chronic disease population. Oncology patients tolerate a lot. We just don't know in the autoimmune space whether they're willing to tolerate as much. Okay, here also we have a fast track designation that was announced yesterday. So 514, living therapies, as you know, enhanced tissue and lymphoid organ penetration versus monoclonal approaches. Our product has a favorable safety profile, deep ablation of inflammatory B and T cells, and we believe that the potential lays there with a single dose of cure in these particular patients. And we've seen in the oncology side, at least, very durable CRs in our patients. So with that, this is our overall expected milestones I had mentioned. 2024 is going to be a very active year. for us. We already have 2 ongoing clinical programs. We have a preclinical program that's going to hopefully make it to an IND stage also. We're close to candidate nomination, but we are excited to have that potential for patients to have durable responses and the potential for cure, which is what all of us want. Thank you very much for your time and appreciate the attention.